So let's talk about the chylomicrons individually. So the chylomicrons, which is the first type of lipoprotein I discussed, and to actually transfer the dietary fat we eat, and when this fat goes through the intestines, and these proteins surround the triacylglycerides. The chylomicron is very high in TGs. It's very high. So it's the largest in size, lowest in density. So first stage is the formation of this initial chylomicron. We call it the nascent chylomicron. The first thing that is gonna happen when this lipid gets surrounded by lipoprotein, the formation of this chylomicron will start by we have first the structural apoprotein. It's very important. It's fixed. It's always there. The B100. The B100 is unique for the chylomicron. So you have to know this. The B100 is unique for the chylomicron. It's structural. It's always there. So this is the nascent form. This is the initial form of the chylomicron. The second stage is formation of the mature. How does the chylomicron, the initial stage, gets into becoming a mature chylomicron? The HDL, as I said, the healthy one, the high density lipoprotein, highest in proteins, have actually ApoC and ApoE, ApoC2 of course. The HDL will give off two apoproteins donated to the chylomicron. So the chylomicron now have the structural apoprotein, the B100, it's fixed, and it's going to have the ApoE donated from the HDL and the ApoC2. Each one have its own function, and now we're going to know why did HDL, the high density lipoprotein, actually donated these two. Because the ApoC2 actually is going to activate a very important enzyme, the lipoprotein lipase C. So the ApoC2, when it gets onto the chylomicron, it's actually a mature one, it's a mature form of the chylomicron. ApoC2 will activate the lipoprotein lipase C. And this lipoprotein lipase C is very important for metabolizing the triacylglyceride inside the chylomicron. It's going to metabolize, it's going to break it down into fatty acids and glycerol. This is very important for activating the lipoprotein C, the lipoprotein lipase C, and the lipoprotein lipase will go and metabolize the triacylglyceride inside the chylomicron. The second is the ApoE. Why is it important that the HDL donate ApoE and ApoC? Okay, I know ApoC2 is important for activating an important enzyme. ApoE is important for when this chylomicron reaches the liver is going to recognize the ApoE, and that's how the chylomicron is going to be taken up by the liver. So you can see the big picture here. The chylomicron initially is just a nascent chylomicron. It have B100, which is fixed structural apoprotein for the chylomicron. It's highest in fat, highest in lipids, so it's very large, least in protein, so the least dense. And then the HDL gives off the apoprotein E and the ApoC2, because ApoC2 is important for metabolizing the triacylglyceride inside the chylomicron. Why? ApoC2 is going to activate the lipoprotein lipase C that's gonna break down the triacylglyceride. ApoE is there because it's important for recognizing of the liver receptor to this ApoE. So the liver is going to recognize ApoE and take this chylomicron inside. So this is the first two stages of formation of the chylomicron, the nascent and then the mature. The mature, how does it get matured? Because HDL is going to give off these two apoproteins. The third thing, the formation of remnant chylomicron. So after ApoC2 activates the lipoprotein lipase enzyme, the lipoprotein lipase enzyme is gonna break down the triacylglyceride into 3 fatty acid plus 1 glycerol And the fatty acid is going to be delivered to the tissues for energy, and the one glycerol is going to actually used in the tissues for glycolysis, gluconeogenesis, and whatever. So the lipoprotein lipase C is very important in breaking down the triacylglyceride inside the chylomicron. Where does this lipoprotein lipase C, where is it? It's actually anchored in the endothelial cells, in the arterial walls of the vessels. It's always there by negatively charged proteoglycans. So this is actually on the cell walls and the cell membrane, outer portion of the cell wall of the endothelium in the blood vessels. It's always sitting there, so when the chylomicron is just walking around with its ApoC2, is going to find the LPL, and the C2 is going to activate it and form the and metabolize the triacylglyceride. So this is the remnant chylomicron is formed after the LPL actually metabolizes the triacylglyceride. Now, after we broke down our triacylglyceride inside the chylomicron, the liver is going to uptake this remnant chylomicron. So the question is the liver going to uptake the nascent chylomicron or the mature chylomicron or the remnant chylomicron? The answer is the remnant chylomicrons after it gets metabolized by the lipoprotein lipase C. So uptake of this remnant chylomicron by the liver after the ApoC2 activated the LPL, and the LPL actually metabolized the triacylglyceride. And now it's called the remnant chylomicron. Now the liver is going to take it because it has ApoE, which is going to recognize the receptor on the liver, ApoE receptor. So now we're done with the formation of chylomicron and its pathway. Now we get the picture. The chylomicron gets formed, and in the nascent or initial way, the second formation of the mature chylomicron, the third is the remnant chylomicron that happened as a result of the lipoprotein lipase C metabolizing the triacylglyceride. The fourth is taken up by the liver. So now we're done with the chylomicron. I want to talk a little bit about the lipoprotein lipase C. It's very important. So this enzyme, it's activated by ApoC2, as we said. So this enzyme is actually attached on the luminal capillaries of most tissues, except, so it's there in most tissues. This enzyme is there in most tissues except for the brain and the liver. So the brain and the liver is the places where we don't have the lipoprotein lipase C. And the lipoprotein lipase C is activated by the ApoC2. But insulin, when we have insulin, we just ate in a fed state. We have insulin and insulin there to let glucose go inside the cells and whatever. The insulin is going to help the lipoprotein lipase C get stimulated. So the insulin wants the triacylglycerides to be broken down. So insulin is going to stimulate this enzyme. It's very important. So let's talk again about the functions of the lipoprotein lipase. It hydrolyzes the triacylglycerides, break it down into fatty acids and glycerol. As I said, the fatty acids are stored in the adipose, used for the energy, adipose tissue, used in energy for in the muscles. And the glycerol is transferred to the liver, and we can have glycolysis, gluconeogenesis, whatever we can use with the glycerol structure. Now, what happens if we have a deficiency in the lipoprotein lipase? What happens is, so we have two cases where we have deficiency of lipoprotein lipase. The first case in a diabetes mellitus. Diabetes mellitus is when we have decrease of insulin. And I said insulin is important for stimulating lipoprotein lipase. Why is it important for insulin to stimulate the lipoprotein lipase? Because if we recall, I said the lipoprotein lipase is on the walls of the endothelial cells. The insulin stimulates the lipoprotein lipase and helps this lipoprotein lipase goes to the surface of the cell from the inside. And if we have a decrease in the insulin, we're going to have no stimulation of the lipoprotein lipase. So we're going to have increase in the chylomicron. No one is metabolizing the chylomicron. We have increase in the blood. So we need the metabolism of the chylomicron so it can be remnant chylomicron, therefore it will be taken up by the liver. But if we do not have lipoprotein lipase, in case of diabetes mellitus, a decrease in insulin, no stimulation of the LPL, we're going to have increase in the chylomicron. You have to understand this. It's very important. It's like a story. Take it in a fun way and you will love it. The second thing is, second situation where we have a decrease or a deficiency in the lipoprotein lipase is genetically or hereditary. We have deficiency of the lipoprotein lipase C, or we have an deficiency in the ApoC2, not specifically on the enzyme itself, but the one activating the enzyme. ApoC2, I said it's very important, ApoC2, because it activates the LPL-C. So if we have, in these two cases, the diabetes mellitus and the deficiency of the enzyme itself, or ApoC2, the activator, it's going to cause, which is the second disease we're going to have today. So the first disease was the atherosclerosis. We discussed that. The second thing is type 1 hyperlipoproteinemia. Type 1 hyperlipoproteinemia is caused by deficiency of lipoprotein lipase or ApoC2, or in diabetes mellitus, decrease in insulin. This is the second disease we have, the familial lipoprotein lipase deficiency. We're going to have high in chylomicron, because no metabolism by the LPL-C results in hypertriacylglyceremia. So chylomicron, and the chylomicron contains TGs, so hypertriacylglyceremia. The thing here is, at one point, the gimfibrozil is actually a drug that is a lipid-lowering agent that will activate the LPL to decrease the level of chylomicron. So we use this to treat the hypertriacylglyceremia or type 1 hyperlipoproteinemia.